1,000 genes are malfunctioning in your body right now. You won't notice most. But what if you could silence them? Meet antisense oligonucleotides. They're like molecular duct tape. You can silence genes. But how? It's happening in your body right now. Your cells are listening. Are you? For instance, consider the case of Spinraza, a medication that uses antisense oligonucleotides to treat spinal muscular atrophy, a devastating disease that affects 1 in 10,000 babies born in the US. By targeting a specific gene, Spinraza has been shown to improve motor function in patients, with some children even gaining the ability to sit or walk. What's more surprising is that our bodies have a natural process called RNA interference, which can also silence genes, and it's been happening in plants and animals for millions of years. Scientists are still uncovering the intricacies of this process, but one thing is clear: understanding how to harness it could revolutionize the way we approach genetic diseases. You might be wondering, what does this mean for you? The potential is vast, and researchers are working tirelessly to develop new treatments that can target the root cause of diseases, rather than just the symptoms. Your cells are already using this technology, and now it's time for you to learn more about it. Here's what nobody tells you about antisense oligonucleotides. They're like a game of molecular Lego. You build a strand of nucleotides that binds to a specific gene. It's like a key fitting into a lock. The gene is silenced. But what's the lock? It's your RNA. You have 2.4 million RNA molecules per cell. That's a lot of locks. For instance, consider the work of scientists at Ionis Pharmaceuticals, who used antisense oligonucleotides to treat spinal muscular atrophy, a devastating genetic disorder. They designed a strand that bound to a specific gene, increasing production of a crucial protein, and it worked. Patients saw significant improvements. You might be surprised to learn that antisense oligonucleotides can also be used to treat complex conditions like cancer. In fact, did you know that a specific type of RNA, called microRNA, plays a role in regulating gene expression, and scientists have found that certain microRNAs are associated with specific types of cancer? It's a complex process, but essentially, antisense oligonucleotides can be designed to target these microRNAs, potentially leading to new cancer treatments. You're essentially hacking into your own cellular machinery. You might be wondering how this molecular handshake works. Well, it's quite fascinating. In 1978, a scientist named Paul Zemechnik discovered antisense oligonucleotides, which can bind to specific RNA sequences, essentially silencing the gene. This binding mechanism is crucial, as it prevents the RNA from being translated into protein. You have around 20,000 proteins per cell, which is a staggering number. To put this into perspective, consider the case of spinal muscular atrophy, a genetic disorder that affects the production of a specific protein. Researchers have used antisense oligonucleotides to increase the production of this protein, with remarkable results. In one clinical trial, the treatment increased the protein levels by 2.5-fold in just 6 months. It's like a highly targeted intervention. What's surprising is that our bodies naturally produce antisense oligonucleotides, which play a role in regulating gene expression. You see, the RNA is being translated into protein, and this process is happening constantly. It's a complex dance, with molecules interacting and influencing each other. The oligonucleotide binds to the RNA, and the gene is silenced. It's a delicate balance, and one that scientists are still working to fully understand. Most doctors get this wrong. Antisense oligonucleotides aren't just for silencing genes. They can also activate them. It's like a molecular switch. You can turn genes on and off. But what's the switch? It's a molecule called RNase H, a molecular scissors that cuts the RNA, silencing the gene. For instance, consider the case of Spinraza, a medication used to treat spinal muscular atrophy. It uses antisense oligonucleotides to increase production of a crucial protein, and has been shown to improve motor function in patients. But what if you could cut the wrong RNA? You could activate a disease gene. Surprisingly, researchers have found that some antisense oligonucleotides can even recruit an enzyme called RNase L, which can trigger an immune response. This is a double-edged sword. On one hand, it could be used to fight cancer, but on the other hand, it could also lead to unintended side effects. You might be wondering, how do scientists ensure they're targeting the right gene? It's a complex process involving careful design and testing of the antisense oligonucleotides. By understanding how these molecules work, you can appreciate the intricacies of gene regulation and the potential for new treatments. You're probably carrying a genetic disease right now, and it's not active, but it's there, waiting to potentially affect you or your loved ones. What if you could silence it? Antisense oligonucleotides can do that, and they're a game changer. For example, consider spinal muscular atrophy, a disease that affects 1 in 10,000 births. With antisense oligonucleotides, you can prevent the disease from activating, essentially saving a life. But here's the surprising part. Did you know that some genetic diseases can be triggered by something as simple as a change in diet or environmental factors? It's true. Duchenne muscular dystrophy, for instance, is a genetic disorder that affects 1 in 5,000 males, and researchers are working tirelessly to develop antisense oligonucleotides that can combat it. You could be a carrier of one of these diseases, and not even know it. But with the power of antisense oligonucleotides, you could potentially save a life, or even your own. It's a remarkable technology that's still in its infancy, but the possibilities are endless. Imagine being able to prevent a disease from taking hold, simply by targeting the genetic root cause. It's a future that's not too far away, and it's an exciting one. You've probably heard the claim that antisense oligonucleotides are toxic, but where's the proof? In reality, numerous studies have demonstrated their safety. Take the example of Spinal, an antisense oligonucleotide drug used to treat spinal muscular atrophy. It's been administered to thousands of patients, with remarkably low toxicity rates. Less than 5% of patients experience severe side effects. But what about those side effects? They're rare and usually mild. You might encounter some molecular noise, a phenomenon where your cells receive subtle, irrelevant signals. It's like a whispered secret, barely perceptible. Surprisingly, research has shown that molecular noise can even have a beneficial effect, stimulating your cells to become more resilient. For instance, a study published in the journal Nature found that low levels of molecular noise can activate cellular pathways that promote longevity. So, the next time you hear someone claim that antisense oligonucleotides are toxic, you can set the record straight. The evidence simply doesn't support it. You now have a more nuanced understanding of these versatile molecules and their potential to revolutionize treatment options. In 1953, two scientists, James Watson and Francis Crick, made a bet that changed everything, sparking a revolution in genetics. They bet that DNA was the genetic material, and this discovery paved the way for major breakthroughs. But what if they were wrong? What if RNA was the genetic material? This idea may seem far-fetched, but consider the case of antisense oligonucleotides, which can target RNA to silence disease-causing genes. For instance, a company like Biogen has used this technique to develop a treatment for spinal muscular atrophy. With impressive results, in clinical trials, 60% of patients showed significant improvement. But what if you could target DNA? You could silence genes forever, which sounds like a dream come true. However, the catch is that you could also activate disease genes forever, and that's a terrifying prospect. Surprisingly, research has shown that certain genes can be reactivated even after being silenced for generations, a phenomenon known as genetic memory. You might be wondering, what are the implications of this discovery? The truth is, the possibilities are endless, and the consequences are still unknown, leaving you to ponder the potential risks and benefits. You can silence genes with antisense oligonucleotides. But what's the next step? Can you silence disease genes forever? The answer is yes. Take the example of Spinal, a drug used to treat spinal muscular atrophy, which works by targeting a specific gene. In clinical trials, it was shown to increase the levels of a crucial protein by 2.5 times, significantly improving the lives of patients. But what if you could silence the wrong gene? You could create a new disease. Surprisingly, scientists have discovered that some genes have a backup function, where another gene takes over if the first one is silenced. For instance, researchers found that when they silenced a gene responsible for a certain type of cancer, another gene stepped in to promote the growth of the cancer cells. You could cure a disease if you silence the right gene. The choice is yours, but it's not a simple one. You need to carefully consider the potential consequences of silencing a gene, as it can have far-reaching effects on the body. What if the silenced gene has an unexpected role in a different part of the body? The possibilities are endless, and the decision is a complex one.